Today we are all going to learn a new topic, that is here mitosis. Okay. So, what is the mitosis? The term mitosis first introduced and coined by Sir Fleming. The name of the scientist was Fleming. Okay. In 1882, he introduced the term is here mitosis. அப்படிங்கிற term வந்து first introduce பண்ணார். Okay ma? So, what is the mitosis? A mitosis is a type of cell division. Okay. So, where which is takes place? எங்க அது நடக்குது அப்படினு பாத்தீங்கன்னா, that is takes place in the somatic cells. It means that is a body cells அப்படினு சொல்லுவோம். Okay. It is takes place in the root and shoot apex. Because in the root and shoot apex, it containing the meristematic tissues. Okay. So, what is the meristematic tissues? The young and perpetual tissues are present in the root apex and shoot apex. So, the cell divisions occur in these regions. Okay. So, this is the mitosis cell divisions. Okay. So, that is takes place in the somatic cells. Somatic cells abdina, that is called by the body cells abdin solrom. And next one, this is a equational divisions. The mitotic division is otherwise called, that is a equational divisions, because the number of chromosomes in the progeny cell and the parent cell is the same. So, that is known as a equational divisions. Okay. So, the mitosis takes place in the diploid cells. So, what is a diploid cell? That is a 2n diploid cells in the organisms. Okay. But haploid, this is also occur in the haploid cells of some lower plants. Okay. Usually that is occur in the diploid cells, but it is also occur in the haploid cells of some lower plants. Okay. So, this is the, so mitosis is a just introduction. So, next we have to see the two type of mitosis. One is the closed mitosis, another one is here open mitosis. So, in the closed mitosis, so the nuclear membrane is remain intact. Okay, it will not separate. Okay, but in the open mitosis, the nuclear membrane is formed in the separate chromosome. Okay, it has a two daughter chromosome. The two daughter chromosomes is enclosed by the two nuclear membrane. Okay, so here the closed mitosis, the nuclear envelope is remains intact, and the chromosome is migrate towards inside the cell. Okay, inside the nucleus. So actually, there is a chromosomes migrate to opposite pole of the spindle within the nucleus. Okay, how the chromosomes are separated? The chromosomes are separated with the help of the spindle fibers. Okay, the spindle fibers separate the chromosomes to the migrated towards the opposite poles. Okay, so that is takes place inside the nucleus. Okay, nucleus kulleye adu nadakkum. But the cell membrane is remain intact. It will not separate. So that is known as here closed mitosis. So, what are the examples for the closed mitosis? That is a eukaryotic organism including the yeast. Okay, yeast is here. So, unicellular organism and slime molds. Slime molds is belongs to the fungi. Okay, so that is a many single celled eukaryotic organisms. Example eukaryotes, and that is here unicellular fungi or example for that. So, what is a open mitosis? Open mitosis, the nuclear envelope breaks, and the nuclear envelope break agala, but here the nuclear envelope is break, and they have to form a two nuclear membrane into the two daughter cells. If you it has a open mitosis here. In the prophase stage la paakumbodhu, it is covered by a nuclear membrane, it is disappear. This we will see later. So, here that is we can see the two nuclear membrane. So, finally, a cell is divided into two daughter cells. The two daughter cells are covered by the nuclear membrane. Okay. So, that is a open mitosis. The envelopes, nuclear envelope breaks down and then reforms the two sets of separate chromosome. The two sets of separate chromosomes are covered by the nuclear membrane. That is the most of the plants and animals. Okay. So, now we are going to see the mitosis, stages of mitosis. Okay, so what are the stages in the mitosis? The mitosis consists of 4 stages. The first one is a prophase, and second one is a metaphase, and another one is here that is a anaphase, and last one that is a telophases. It has 4 stages in the mitosis. We will see one by one. That is a prophase. Okay, the prophase is the initial phase of the mitosis. Okay, so it has this is the longest phase compared to others. This is a longest phase. So, in the plant cells, so what are the process takes place in the prophase. Prophase la ennalam nadakkum appadina, the first nuclear membrane will be disappear and nucleolus will be disappear, and end of the prophase and some of the cell organelles also disappear. Example for mitochondria, endoplasmic reticulum, these are all disappear. And what will happen in the prophase? Okay, appo edella vandu appear agum appdin pathina, the spindle fibers are formed, okay, in the initial stage of the prophase. So the plant cells initiations of the spindle fibers, okay, the spindle fibers are initiated here, okay. Nucleolus will be disappear, not only nuclear membrane, nucleolus. What is the nucleolus? In center of the nucleus we can see a prominent structure is called nucleolus, okay. The nucleolus will disappear during this stage. Nuclear envelope breaks down, it will disappear, okay. And Golgi apparatus, the endoplasmic reticulum, these are all also will disappear in the end of the prophase. So what about that spindle fibers? How the spindle fibers are formed in the plant cell and the animal cell? Okay, in the animal cells have a centrioles. Okay, so they have to form the spindle fibers. The centrioles are gives rise to microtubules. From the microtubules, so the radiating arrangement of the microtubule is known as asters. Okay, so from that asters, the spindle fibers are formed. This is happened in the animal cells. So what about in the plant cells? In the plant cells, the centrioles are absent. Okay, appo centrioles illana, how the spindle fibers are formed? The spindle fibers are formed without centrioles, so that is in outside of the nuclear membrane. Okay, so they have to form a spindle fibers in the plant cells without the centrioles. So the animal cell, the centrioles extend, okay, the animal cell pathina, centrioles they have to extend an array of the microtubules, okay, it gives rise to the microtubules, and the microtubules are reach pole of the cells, okay, rendu pole irukku and the rendu pole ku vandu reach agudhu. This arrangement of the microtubules are called asters, okay. Actually the radiating of the microtubules அப்படியே அந்த விரிஞ்சு வரக்கூடிய அந்த இதுக்கு தான் என்ன பேருனா asters அப்படினு சொல்றோம். The plant cells do not form the asters. Okay, our plant cells asters illana, how it is formed? The spindle fibers are formed in the outside of the nuclear membrane. Okay, so here the spindle fibers are made up of. So what is the spindle fibers? The spindle fibers are made up of, that is here 3% of RNA and 97% of tubulin. Okay, so it is made up of 97% of tubulin. So then only they have to stretches. Okay, actually that is a normal stretches, they can move up to the pole, and the pole எவ்வளவு தூரம் இருக்கோ அது வரைக்கும் தான் அது stretches பண்ண முடியும், அதுக்கு மேல stretch பண்ண முடியாது. ஓகேவா? So that is the nature of the spindle fibers. So what about in that? Some metaphase. Okay, so இப்போ metaphase அப்படிங்கிறது next stage of the prophase. அப்போ what happened in the prophase? Prophase-ல என்ன நடந்தது அப்படினா, the nuclear membrane disappear, nucleolus வந்து will disappear, and some of the cell organelles வந்து will disappear. What it is a reappear அப்படினு பாத்தீங்கன்னா, that is here spindle fibers are formed. The spindle fibers in plant cell and spindle fibers in animal cell we studied. So next we have to see the metaphase. Okay, so during the metaphase, the two sister chromatids, you can see here the two sister chromatids, or they have to separate it to each other. So we can see the structure of chromosomes during the metaphase. Okay, metaphase is a very important phase. So we can see the structure, shape and size. Okay, a number of chromosomes we can clearly studied during this period. Okay. So this is a chromosome is consists of two sister chromatids. The two sister chromatids are attached to the centromere. We know very well that is attached to the centromere. Okay. So what is in the centromere? The centromere consists of a complex system of kinetochore. Okay, so protein complex that is in the kinetochore in the centromere. So the kinetochore is attached to the spindle fibers. Okay, so where is the kinetochore? The kinetochore is present in the centromere. So this is the centromere region. Okay, so this is a centromere region. In the centromere region we can see the kinetochore. Okay, the kinetochore is attached with the spindle fibers. Actually the spindle fiber one end is attached in the equatorial regions. This is the equator end, and another one is a pole end. Okay, spindle fiber oda oru end vandu equator layum, another end vandu it is attached to the pole oda attached ஆயிருக்கு. So where which is attached to the equator end, that is known as a positive end. Okay, which end is attached to the pole, that is known as a negative end. Okay, இப்போ ஒரு positive end ஒரு negative end இருக்கு. Spindle fiber இங்க attached ஆகிறதுக்கு என்ன reason? Because the presence of the kinetochore in the centromere. Okay. So of the centromere, the spindle fibers are made up of tubulin. Already I said the spindle fibers are made up of tubulin, okay, and RNA, and most of the ratio on the tubulin is present. So next, what is the important process in the metaphase? In the metaphase, so all the chromosomes are, so they have to come close to each other and compact, they have to, so arranged on the equatorial regions, okay. So எல்லா chromosomes equatorல வந்து arranged ஆயிருக்கு அப்படினு சொல்றோம். So the arrangement of the equator, so all the chromosomes on the equator region, so this arrangement is known as here equatorial plate அப்படினு சொல்லலாம், இல்ல metaphase plate. Okay, so all the chromosomes are arranged in the central, the middle of the chromosomes, so that the middle regions of the cell. Okay, so that is known as a metaphase plate, illana equatorial plate. So this is what about in the animal cells and plant cells. So in the plant cells, the chromosomes are irregularly arranged. Okay, எப்படி arranged ஆகுமா? So ஒரு பெருசு சின்னதெல்லாம் கிடையாது, they can smaller chromosomes and larger chromosomes also there, so they have to arrange irregularly. But what about in the animal cells? In the animal cells, the smaller chromosomes are arranged in the center region and the larger chromosomes are arranged in the peripheral regions. Okay ma, அப்ப சின்னதெல்லாம் சென்டர்ல இருக்கும், லார்ஜர் எல்லாம் அங்க இருக்கு பெரிஃபரல் ரீஜன்ஸ்ல அரேஞ்ச்டா இருக்கு. So this arrangement is called the equatorial plate arrangement, otherwise metaphase plate arrangement. Okay, the stage where the chromosomes or morphology can be easily studied. Okay, we can study the morphological structures. So what is a morphological structure? That is the external appearance. Okay, we can see the size, shape, structure and number of chromosomes we can see during this period. So here the kinetochore, already I said the kinetochore is made up of the protein complex. Okay, that is two kinetochores in the centromere, that is they have to form along with the arms. Okay, the protein complex present in the centromere where the microtubules are attached in the trilaminar disc shape. This is known as a trilaminar disc shape. Okay, so where is the centromere? So this is this region is known as a centromere region. Okay, in the centromere we can see the kinetochore. So this is the kinetochore. So this is the inner kinetochore and outer kinetochore. Actually two kinetochores, a pair of kinetochores in the centromere. Okay, so this kinetochore is made up of protein complex. So this is attached with the microtubules. This is the microtubules. Upon the microtubules edoda attached aayirukku, this is attached to the kinetochore. Okay, so the microtubules gives rise to the spindle fibers. Okay, so another end of the spindle fiber is attached to the, that is a pole. Okay, one end is attached to the kinetochore and another end is attached to the opposite poles. Okay, so this arrangement is known as a trilaminar disc-like plate. In the arrangement as outer kinetochore, inner kinetochore, இதெல்லாம் சேர்ந்து ஒரு plate மாதிரி ஒரு trilaminar. You can see here trilaminar like structures. Okay, மூணு layers இருக்கும். It has a 3 layers. So that is known as a trilaminar disc-like structure is there. So next we have to see the anaphase. Okay, so the anaphase is a very important phase in the mitosis. In the anaphase we can see the movement of the chromosomes. Okay, we can see the clear migrations of the chromosomes. Okay, so we can see the different shape of the chromosome during the anaphase. Okay, when the chromosomes are separated from each other and moving towards the opposite pole, so we can see the different structure. We can see the L-shaped, V-shaped, so and I-shaped structure we can see. That is here metacentric, submetacentric and telocentric, another one is here acrocentric. Okay, so based on the position of the centromere we can classify the chromosomes. So that classification, that morphological structure we can see in the during this phase. Okay, so the anaphase, each chromosome splits. Each chromosome should be split. How can they split? In the centromere. Even the centromere also split into two. Okay, ma'am. Each chromosome split simultaneously and two daughter chromatids begin to migrate towards the opposite poles. Actually, so this is a structure like that. So this is your real structure of the chromosomes. In the chromosomes they can split. Okay, in the centromere. Now half of the centromere along with the daughter chromatids. So another pair is along with the daughter chromatids. Okay, அப்ப half of the centromere வந்து என்ன ஆயிடும்னா they can split along with the kinetochore also. Okay, so this is the splitting arrangement. அப்ப என்ன ஆகுதுன்னா half of them is move towards the opposite pole and another half move towards the another opposite poles. So this is the towards opposite poles of your cell. So what is in the APC? APC அப்படிங்கிறது என்ன அப்படின்னா this is very very essential for the movement of the anaphase. Anaphase ரொம்ப முக்கியமான ஒரு protein complex. ஓகேவா? So anaphase promoting complex. Anaphase வந்து promote பண்ணக்கூடியது, trigger பண்ணக்கூடியது. It's very important for the involvement of, that is the movement of the chromosomes towards the opposite pole. So a cluster of proteins. So what is in this region? A cluster of proteins are present. It induces the breaking down of cohesions of protein which leads to the separations of the chromosomes. Actually it has the cohesions of proteins. Where is the cohesions of proteins? So kinetochore. Kinetochore is made up of a protein complex. So on the protein complex it will be separate. Okay, and the protein complex is separate பண்றதுக்கு தான் நம்ம என்ன பண்றோம் அப்படினா this is a protein complex. It will separated with the help of the, so that is your APC, that is your anaphase promoting complex. and the cohesions ஒன்னோட ஒன்னு இருக்குறத என்ன பண்ணுது அப்படினா it will be separated, detached from each other. Okay, so that is a breaking down of cohesion proteins which leads to the separation of the chromatids during the mitosis. Okay, during the mitosis that chromatids will separate, the sister chromatids will separated from each other. Okay, so this is the transition of metaphase to, that is here anaphase in conditions. Okay, இந்த டைம்ல தான் என்ன ஆகுது அப்படினா டோட்டலா anaphase-க்கு change ஆகுது. metaphase-ல இருந்து totally that is changed into anaphase in condition. Okay, in the anaphase the centromere splits longitudinally. இப்படி இருக்கிற அந்த centromere they have to split longitudinally. Okay, so the centromere splits longitudinally along with the kinetochores, the two sister chromatids from each other. So when the sister chromatids separate, the actual partitions of the replications of genome should be complete. Okay, so when the chromatids or sister chromatids are separated, so the totally separated actual partitions. Okay, so actually ரெண்டுமே partition ஆயிரும். So whatever that is molecules, that's a components, okay, that's a cell organelles also will synthesize, not now in the telophase in conditions. Okay, so in the anaphase is a very important phase. So we can see the different shape of the structure. Already I said that's a V-shaped, L-shaped. V-shaped, that's a metacentric. So L-shaped that is a submetacentric, and the telocentric and the acrocentric, that's a rod shape chromosomes we can see during this period. And next we have to see the telophase. Okay, this is here, so last phase of the mitosis. In the telophase, so the both chromatids are totally separated from each other. Actually the karyokinesis will be over. Okay, inga da karyokinesis namakku nadakku. Okay, so you can see here, so that is the cleavage between the two daughter chromatids. It's a total, that is a cytokinesis. Okay, so first we can see the karyokinesis, followed by cytokinesis also occur during this period. Finally we can see the two daughter cells. Okay, in the telophases, so whatever is disappear in the prophase, so that material will be reappear in the telophases. So edalla disappear aachu appdin pathinga appadina, so nuclear membrane disappear during the prophase, it will reappear. Okay, nucleolus will reappear, and some of the cell organelles also will reappear during this period. Okay, so what is a karyokinesis? The division takes place in the nucleus is known as a karyokinesis அப்படினு சொல்றோம். ஓகேவா? So அதுக்கு அடுத்து the division takes place in the cytoplasm, that is known as a cytokinesis. So the two sets of daughter chromosomes reach opposite pole of the cell and mitotic spindle fiber disappear. Okay, so actually the mitotic spindle fiber is appear during the prophase, so here that is disappear. Okay, and divisions of the genetic materials completed. So all the genetic materials in the nucleus will be completed, so during the karyokinesis. Okay, so followed by cytokinesis. Cytokinesis means that is a divisions in the cytoplasm. You can see here, so there is a cytoplasm. Okay, also it's a divide. Okay, they have to divide to one another, they have to form a two daughter cells. So nucleolus will, that is here already nucleolus disappear during the prophase, but nucleolus will reappear. So how the nucleolus will reappear? So because the presence of the secondary constrictions. Okay, so that is known as here, that is here nucleolar organizers. நம்ம chromosome structure படிக்கும்போதே பார்த்திருக்கோம். So nucleolar organizer எது அப்படினா, that's a secondary constriction. So with the help of the secondary constriction, nucleolus will reappear during the telophase. So nuclear membrane and everything should be reappear and formed around each set of the chromosome. Now the chromosomes are decondensed. The chromosomes என்னாகும் அப்படினா, that is elongated. So decondensed வந்து ஆயிரும். ஓகேவா? So they have to form a initial stage. Initial stage ல எப்படி ஒரு thread மாதிரி இருந்துச்சோ அந்த மாதிரி அது form பண்ண ஆரம்பிச்சிரும். So between that in plants, okay, how the divisions takes place in the plant cell, how the cell wall is formed between the daughter cells in the plant cell and in the animal cells. Okay, இப்போ cytoplasm division ஆயிருச்சு அப்படினா, so the both of them will separated, so they need a separate cell wall and cell membrane. How it is formed in the plant cells? Okay, actually in the plant cells the cell wall is formed with the help of the phragmoplast அப்படினு சொல்லுவோம். What is a phragmoplast? Actually a small, that's the fragments are there. Okay, the fragments is like a vesicles. Actually the vesicles are formed from that is derived from the Golgi apparatus and the endoplasmic reticulum. So adula irundhu varakudi and the small fragments da, that's a phragmoplast அப்படினு சொல்றோம். So they have to form a cross wall. Actually the cross wall is formed in the plant cells from center to the margin. Okay, but what about in the animal cells? From the periphery to the center, that's a cell furrow in conditions. But cell plate la pathina center to margin they have to form. Okay, and finally the middle lamella is formed between the two cells. The two cells are separated. Each cell they have to build the cell wall and cell membrane, and the middle lamellae is formed. Okay, middle lamellae form ஆன உடனே they have to separate it. In plants phragmoplasts are formed, so between the daughter cells. So எங்க form ஆகும் அப்படினா, this is a phragmoplast. The phragmoplast is nothing, actually that is your vesicles. Okay, vesicles of that is your endoplasmic reticulum. Okay, the cell plate is formed between the two daughter cells, reconstructions of the cell wall takes place, and finally cells are separated and distributions of the cell organelles. Okay, so ரெண்டு பக்கமும் அப்படியே separated ஆகும்போது cell organelles also equally distributed. Okay, இங்க என்ன cell organelles இருந்ததோ அதே cell organelles இன்னொரு செல்லையும் இருக்கும். They have to equally distributed into the macromolecules into newly formed daughter cells. So what is the cytokinesis? The cytokinesis in animal cells, that is a cell furrow அப்படினு சொன்னேன் இல்லையா? Cytokinesis அப்படிங்கிறது the divisions in the cytoplasm. So this is a cell furrow in condition, that is from the periphery to the center. Okay, the outer cell வந்து என்ன ஆகும்னா, they have to move towards inward. Okay, inward ஆ வந்து move ஆகும். So there is a cleavage in the furrow methodல வந்து cleavage ஆகுது. Okay. So this is a cell furrow in conditions. So in the animal cell, so that's a contractile process. Contractile அப்படிங்கிறது அப்படியே என்ன ஆகும் அப்படினா நல்லா contract ஆகி சுருங்கி, so from the periphery to the center, the cell wall, okay, the cell membrane should be contract. So அந்த contract ஆகும். So they can divide it into two bundle. Okay, the ring consists of a bundle. The ring அப்படிங்கிறது தான் அந்த contract and the furrows அப்படினு சொல்றோம். So that is a bundle of microfilaments assembled from actin and myosins. Okay ma? So when we can see the contractile process, so actin, myosin is nothing, that is a protein complex. Okay, the both of them is protein, both of them is a filaments. So this is very important for the separations of the chromosomes. Okay ma? So actin, myosin ரெண்டுமே is used for the separations of the chromosome. The actin is always associated with the myosin. Myosin ஓட சேர்ந்து தான் அது வந்து act பண்ணும். Actually myosin is very important. So myosin, they have to transform the chemical energy into, that is a mechanical energy. Okay, so what is the chemical energy? In the form of ATP. Okay, so the ATP energy வந்து, so mechanical energy, because they both of them is divided into each other. Okay, ரெண்டு மூவ் ஆகணும் இல்லையா? The both of them detached from each other. அந்த mechanical energy கொடுக்கக்கூடியது பார்த்தீங்கன்னா myosin. Okay, myosin is also one of the protein filament, so which is convert the chemical energy in the form of ATP is converted into mechanical energy. The both of them is very important for that, for cytokinesis in animal cells. The fibril generates a contractile force that draws the ring inward, forming a cleavage. Okay, the ring அப்படிங்கிறது மேல இருந்து ஃபார்ம் ஆகக்கூடிய இந்த ring. This is a cleavage furrow அப்படின்னு சொல்றோம். So due to that microfilaments, okay, microfilaments அப்படி contractile process ஆகுறதுனால தான் ஒரு single cell வந்து ரெண்டா divide ஆகுது அப்படின்னு சொல்றோம். So this is divides the cell into two. Okay, so cytokinesis in plant cell is the divisions of, that is a cytoplasm often starts during the telophase. Telophase அப்பமே என்ன ஆகும் அப்படினா, so அந்த plant cells வந்து divide ஆகுது. Plant cell ஓட division that is takes place from center to margin. But what about in the animal cell? That is here margin to center. Margin இருந்து சென்டருக்கு வரும்போது furrow வரும். But what about in the plant cells? The cell plate is formed in the center of the cell. Okay va, the cell plate eppadi form aguduna, because the presence of the vesicles in the Golgi apparatus and endoplasmic reticulum. So in the plant cell, the plate grows from the center, so towards the cell wall. Okay va, center la irundhu enga form agudhu appadina, so there is a form from center to, okay, so this is the center. In the center la irundhu da enna agum appadina, this is formed from towards the lateral walls. Okay, the chinna chinna fragments irukku, this is a phragmoplast. Fragments, that is called a phragmoplast. The phragmoplast is, that is a vesicles from the Golgi apparatus and endoplasmic reticulum. It's like a small bricks. Okay, half bricks eppadi irukumo adha mari idu vandu form agudhu. So and finally they have to divide it. Okay, finally it will divide into each other. Rendum divide ayirum. So during this period the middle lamellae is formed. So the microtubules of the phragmoplast move the equator, fuse to form a new plasma membrane, and the materials which are placed there becomes a new cell wall. Okay, so what is the microtubules? Already we said earlier, microtubules is used for separations of the chromosome, because which gives rise to the spindle fibers. So adu ellame enna aduna, after phragmoplast irukkudiya the microtubules vandu move towards the equators. Okay va, so the equator la move aagi anga irundha from the equator to, that is here another peripheral. Equator la irundhu poles, from that is a center to periphery they have to form. So and the materials which is placed there becomes a new cell. Inga da enna aguduna, new cells vandu form agudhu. Okay, the first stage of the cell wall construction is dividing the newly forming cells are called the cell plate. Okay, that is a equatorial plate appdin solrom illaya, adhe mari the first form agakuriya and the phragmoplast இதை வந்து என்ன சொல்லலாம், that is a cell plate. We consider this is your cell plate. The cell plate eventually stretches. They have to stretches towards the periphery region and they have to form a middle lamellae. Okay, so the cellulose build up on each side of the middle lamellae. So what is the cellulose? Cellulose is the cell wall material. Okay, the basic cell wall material எதுனா cellulose தான். Okay, so the cellulose is build up each side of the middle lamellae. Actually middle lamellae is made up of the calcium and the magnesium pectate. So middle lamellae vandu or cell wall material is different, but this is act as here, there is a cement. Okay, this is present in the middle region of the cell wall. So primary wall ko secondary wall ko center la irukakudiyada enna solra, it's a middle lamellae. So cellulose is built up on each side of the middle lamellae to form a cell walls into new. So next we are going to see the significance of mitosis. So what are the significance of mitosis? The main thing is, so they have to repair the tissues. Okay, if you have any wounding in your body, so it will repair, it will regenerate with the help of the, so that is a mitosis. There is a genetic stability. Okay, genetic stability they have to maintain, because the number of chromosomes in the parent cell will maintain in the daughter cells. So adanal enna solrom, that is a genetic stability. Genetic vandu maintain pannudu. And growth, so all the multicellular organism they can grow. Okay va, so that is even the small plant they can grow as a tree because the presence of cell divisions. Okay, that is a mitotic cell divisions. The meiosis takes place in the, so that is in the meiosis in the reproductive cells. Okay, mitotic enga nadakum appadina somatic cells la vandu nadakudhu. The number of cells making up the tissues increase. Okay, tissues oda size vandu increase pannom. The new cells must be identical to existing ones. Okay, we cannot see any different cells. So actually whatever in the parent cell we can see in the daughter cells. Okay, exact adhe dhaan irukum. So repair of tissues அப்படிங்கிறது ஏதாவது damage, if you have any damage in your body, ஏதாவது crack ஆயிடுச்சு இல்ல wounding ஆயிடுச்சு அப்படினா it will recover with the help of the mitosis. And asexual reproduction. So asexual reproduction is your, actually this is a clone method அப்படினு சொல்வோம். So that any part of the plant body which gives rise to the entire organism, because that is involved in the cell divisions. ஒரு buddo இல்ல ஒரு root or tubers or anything you have to detach it from the plant body, so it will grow and gives rise to the entire plant. say asexual reproductions, say especially in the yeast and amoeba. So another one in flowering plant structures such as a bulb, corms, tubers, rhizomes, runners. So these are all the vegetative part. Okay, this is vegetative propagules. The vegetative propagules, they will undergoing the mitotic division to gives rise to the entire plant. Okay, and the divisions enlarge the tissues. Tissues are enlarged. Continuous divisions and the tissues enlarged. Okay, next one the regenerations. Regenerations the arms of starfish or lizard. So in the lizard or the tails cut it will regenerate. So anything. Okay, some of the plants they can regenerate even the whole body. Okay, full body kuda adu enna pannu. So regenerate pannakudiya capacity adukku undu. So this is the significance of mitosis. So shall we recall once again that is mitosis. What is the prophase? First one prophase அப்படினு பார்த்தோம். So prophase la என்ன ஆகுது அப்படினா that is a spindle fibers form ஆகுது and nuclear membrane, nucleolus and all the cell organelles will disappear, isn't it, in the prophase. So what about in the metaphase? So all the chromosomes are arranged on the equatorial regions. Okay, so all the chromosomes are attached to the spindle fibers. So one end of the spindle fiber is attached to the kinetochore and another end is attached to the opposite pole. Okay, that is arranged in the equatorial regions. So that is known as a equatorial plate, otherwise that is a metaphase plate அப்படினு நம்ம சொல்றோம். Okay, so during this metaphase we can see the structure, shape, size and number of chromosomes are very clearly visible. We can study the morphological structure of the chromosome. So during the metaphase. Okay, so in the anaphase, so the chromosomes are start to move towards the opposite pole. Okay, so in the anaphase we can see the different shape. So here L shaped, V shaped and that's I shaped, different type of the chromosomes based on the centromere, position of the centromere, we can see the different shape of the structure here. Okay, in the anaphase. And the telophase, what will happen? So then the telophase, that is everything will be, so there is a spindle fiber or disappear and the nuclear membrane, nucleolus, all the cell organelles will be reappear in the telophases. Okay, in the end of the telophase we can see the, so that is a cytokinesis. So cytokinesis in the plant cell is a cell plate method. So cytokinesis in the, so the animal cell there is a cell furrow method we studied. Okay, so thank you children, thank you so much for your cooperation.